When comparing traits across species, well, they can either be homologous or analogous, and it's really important to keep these two distinct. Homologous traits are inherited from a common ancestor. So basically, because of that, you would use homologous traits to construct a phylogenetic tree. You cannot use analogous traits to do that. So let's look at some examples of homologous traits. The wings of birds and the wings of bats happen to be homologous as forelimbs. And the reason for that, as we'll see in a moment, is they have the same underlying bone structure. However, the wings of insects are not homologous with birds or bats. They don't have the same kind of structure, and even though they function the same. And because they function the same, they would be analogous with birds and bats, but not homologous. So, for example, here is an X-ray of a bat's wing. As we can see the bones. And this is not an X-ray, it's just a view from below. But again, the bones are pretty apparent. And, of course, they have similar functions, as do the wings of insects. But the wings of insects, like I said, do not have the same underlying bone structure. So what you're seeing here, the wings of bats and the wings of birds are homologous. So you can use the underlying bone structure as partially involved in reconstructing phylogenetic trees between birds and bats. Obviously, you would use a lot more in addition to that. So again, the bat wing, the bird wing, you can see the bones. They're of course they're different, but they are homologous to one another. Bees, insects, there's no bones there whatsoever. The homeobox genes are intriguing because they are homologous across a very wide range of species because they function by laying out the body plan. Again, in a very wide range of species, insects and, invertebrates in general, as well as vertebrates. However, they function differently depending on the context in which they are found. Even though the genes themselves are identical, they do different things specifically in each species. By the context, I mean the species in which these genes are found. But the genes themselves are exactly the same genes.
Suddenly, the commonality of form among animals was understood. Animals resembled each other because they all used the same set of genes to build their bodies. A set of genes inherited from a common ancestor that lived long ago.
What fascinates modern biologists is that all these different animals don't just look the same. They are using virtually the same set of key genes to build their bodies. The body plan genes determine where the head goes, where the limbs go, and what form they take, whether they are arms, legs, or wings. Another set of genes determines an animal's body patterning, the blotches, the stripes and spots. It is the same genes at work in every creature, from the leopard to the peacock to the fruit fly. And yet they produce radically different results. This has led scientists to a crucial insight about how animal bodies have evolved. It's not the number of genes that counts.
It's not the genes you have, but how you use them that generates the great diversity of the animal kingdom.
So the genes themselves are homologous. So the homeobox genes that you'll find in a fruit fly are homologous to the homeobox genes you'll find in a human. But they obviously operate differently in each species because otherwise the species would look identical, and of course they don't. But the genes themselves, the homeobox genes themselves, are homologous. By the way, sometimes you'll just see homeobox abbreviated as HOX, H-O-X. I've even seen it as H-O-M. It's all the same thing, the homeobox genes. This is the brain of an arthropod. And there's a structure in the brain called the central body. It's abbreviated here CB. So it's this area right here. It's part of the arthropod central nervous system, central body. And here it is again. We see it in color blown up. So all of this that I'm outlining is the central body in the brain of an arthropod. This is the basal ganglia, which is a set of nuclei, and you can see the names of those nuclei. Again, nuclei are groups of cell bodies found inside the brain. The caudate, the putamen, the accumbens, and so on and so forth. They all compose the basal ganglia, which collectively are involved in movement and all kinds of other things as well. And these are just different views of the brain. So all of the elements in color in each of these views are these different nuclei composing the basal ganglia. Well, guess what? It turns out that the arthropod central body and our basal ganglia are indeed homologous. Why is that? Well, it's because, on the one hand, they're derived from exactly the same genes. They mediate inhibitory and excitatory pathways in the arthropod brain and in our brain. And if they become dysfunctional in some way, there's going to be impairments in movement, memory, attentiveness, sleep, and probably some other things as well in both arthropods and us. So they are functioning exactly the same in arthropods as well as in vertebrates, including humans. So those were homologous traits. What are analogous traits? They're traits that are derived from two independent hereditary lines. They're only similar in function or appearance, but they cannot be used in looking at phylogenetic relationships whatsoever. So, as I said before, the wings of birds and the wings of insects would be analogous, not homologous. They function the same way, but other than that, they are totally, totally different from one another. By the way, notice how similar a sphinx moth is to a hummingbird's appearance. Very similar. In fact, if you see them flying around in your garden, sometimes you have to look twice to make sure, if you think you're looking at a hummingbird, to make sure you really are and that it's not a sphinx moth. If you get a close view, you can certainly tell the difference. The chameleon has some interesting eyes, as does the sand lace fish. They both can move independently of one another, as you see in these videos. So the left and right eye do not have to move the same way. They are capable of independent movements. But beyond that, there is no underlying structural similarity. So these are analogous traits between the chameleon and the sand lace fish. Basically, they've evolved independently, probably, I mean, we don't know for sure, but probably because of the similar lifestyles and the various ecological pressures that these animals are faced with. I mean, it really helps both of them because of their ecology to be able to see in front, on the side, behind, with independent eyes. And behaviorally, there's a display that gulls do. A whole variety of gull species do this display. It's called head flagging. And the point that I'm trying to make here is how part of this display involves them turning the back of their heads to one another, even if only for a moment. Well, ducks do something called a head-up, tail-up display, and a component of that is also turning the backs of their heads to one another. So they look similar, but they are analogous between gulls and ducks, not homologous. What is interesting is among gulls, this head-turning display is homologous from one gull species to another gull species. And the head-up, tail-up display in ducks, homologous from one duck species to another duck species. But between gulls and ducks, they are only analogous. So among ducks, for example, you can use this as the basis of looking at phylogenies. Konrad Lorenz did, as a matter of fact. That was one of the many behaviors he looked at in constructing his phylogenetic tree of behavior. But you cannot do it between gulls and ducks. So those are homologous traits and analogous traits.